All right, let's go. Number 10, MC1R, the gene that gave some Neanderthals red hair, and the reason a fraction of modern redheads carry a mutation their species should not have. If you have red hair, or if red hair runs in your family, there's a gene on chromosome 16 that is largely responsible. It is called MC1R, which stands for melanocortin-1 receptor, and it controls how much of two different pigments your skin and hair cells produce. When MC1R works at full capacity, cells produce a dark pigment called eumelanin, which gives brown and black hair its color. When mutations reduce or disable MC1R, the cells switch to producing a reddish-yellow pigment called pheomelanin instead, which is what gives redheads their hair color and their pale, freckle-prone skin. Redheads are a fairly small percentage of the modern human population, roughly 1 to 2% globally, with much higher concentrations in Northern and Western Europe. And for most of scientific history, the assumption was that red hair emerged as a purely modern human adaptation, most likely somewhere in Europe, sometime after our ancestors moved out of Africa into lower sunlight environments. Then, in 2007, a team led by Holger Rumpler at Harvard and the University of Leipzig published a paper in Science that changed that story. The team extracted, amplified, and sequenced the MC1 gene from the bones of two Neanderthals, one from El Cidrón in northern Spain, dated to around 43,000 years ago, and one from Monte Lessini in northern Italy, dated to around 50,000 years ago. They found something unexpected. The Neanderthals carried an MC1 mutation of their own. It wasn't the same mutation that produces red hair in modern Europeans today. It was a different point mutation in a different position within the same gene that produced essentially the same functional effect, reduced MC1 activity, leading to reddish pigmentation. When the researchers introduced this mutation into modern human cells in a laboratory, they impaired MC1 function in exactly the way that leads to red hair and pale skin in living people. Scientists estimated that at least 1% of the Neanderthal population probably carried the mutation, meaning some Neanderthals were, quite literally, redheads. But the mutation Neanderthals used to get there was their own. It was not the same mutation modern redheads carry. And the two lineages appeared to arrive at red pigmentation by two independent evolutionary paths, a striking example of what biologists call convergent evolution, where different populations arrive at the same trait through different genetic routes. The story gets more complicated, though. Later analyses have identified other variants at the MC1 locus that appear to have entered the modern human gene pool through interbreeding with archaic populations. One Val92Met variant at position RS2228479, originally detected only in Neanderthal specimens, has since been found in modern populations, including East Asian groups, and at surprisingly high frequency among Taiwanese Aboriginal peoples, one of the source populations for the Austronesian expansion covered in a previous episode of this series. The full picture is still being worked out. What is not in dispute is that the pigmentation biology of modern humans was shaped, at least in part, by variants that either came from archaic humans directly or that we and they developed in parallel to solve the same problem: how to survive at higher latitudes with reduced sunlight over the same tens of thousands of years. Some redheads today are red because of a mutation their ancestors developed after leaving Africa, and some are red because 60,000 years ago an ancestor of theirs slept with a Neanderthal. Number 9. SLC16A11, the Neanderthal gene that raises the risk for type 2 diabetes for hundreds of millions of people alive today. Type 2 diabetes is one of the most widespread chronic diseases in the modern world. It affects hundreds of millions of people, and its incidence varies dramatically between populations. Some groups develop it at rates far higher than others, even after controlling for diet, lifestyle, modern industrial food supplies, and stress. For decades, the pattern was assumed to be environmental: diet plus lifestyle plus modern industrial food supplies plus stress. Then, in 2014, a large international consortium called the Sigma Type 2 Diabetes Genetics Consortium published a paper in Nature reporting the results of a genome-wide association study across roughly 8,000 individuals of Mexican and Latin American ancestry.
They found a specific gene variant on chromosome 17 that appeared with unusual frequency in this population and that dramatically increased the risk of developing type 2 diabetes. The gene was called SLC16A11. Roughly half of individuals with Native American ancestry carried at least one copy of the higher risk variant. Carrying 2 copies increased the risk of developing type 2 diabetes by an estimated 25%, even after controlling for weight and BMI. Then the researchers compared the human variant to sequenced Neanderthal genomes. It matched. The higher risk version of SLC16A11 turned out to be Neanderthal in origin. It had entered the modern human gene pool through interbreeding somewhere around 50,000 to 60,000 years ago, when the ancestors of all non-African populations mixed with resident Neanderthal populations in the Middle East. It then rose to substantial frequencies in populations that eventually crossed into the Americas. Native American and Latin American populations today carry the variant at frequencies of around 50%. East Asian populations carry it at somewhat lower rates. In European populations, the variant is present at around 10%. In African populations, essentially unmeasurable. Why the variant persisted and even rose in frequency in some populations is not entirely clear. It may have provided some kind of metabolic advantage in the past, perhaps helping ancestors store energy more efficiently in food-scarce environments, that has become disadvantageous only in the context of the modern diet. Whatever the original selection pressure, the result today is that hundreds of millions of people, especially those with substantial Native American ancestry, carry a Neanderthal-inherited genetic variant that increases their lifetime risk of developing one of the most common chronic diseases in the world. The variant is not the sole cause of type 2 diabetes. It's one contributing factor among many, and lifestyle changes remain the most effective intervention. But if you have Latin American ancestry and you have been told your family runs high for diabetes, there is a reasonably good chance that some of the reason is a segment of DNA that walked into your gene pool from a Neanderthal encampment 60,000 years ago. Number 8. BNC2, the pigmentation gene that shapes what your skin does in the sun. If you spend time on a beach in a country where the population has significant Northern European ancestry, and you look at what happens to the people on the beach over the course of an afternoon, you see something specific. Some people tan gradually and evenly. Others turn pink almost immediately, freckle noticeably, and burn if they don't cover up quickly. Some people have almost no visible skin moles at all. Others have many. A substantial part of that variation traces back to a gene called BNC2, basanuclin 2, on chromosome 9. BNC2 is one of the strongest signals of Neanderthal integration identified in modern European genomes. Multiple studies of ancient European DNA have shown that a version of the BNC2 gene inherited from Neanderthals is found at high frequency in modern populations of European ancestry and appears to influence skin pigmentation, tanning response, and the density of pigmented skin moles on the body. The functional effects are subtle but real. Individuals carrying the archaic BNC2 variant tend to have slightly paler skin at baseline, respond to sun exposure with somewhat greater sensitivity, and carry a slightly higher density of pigmented skin moles compared to individuals without the variant. None of this is dramatic. It's not the difference between one person being white and the other being black. It's the difference between the person who tans gracefully all summer and the person who has to reapply sunscreen every 90 minutes and still ends up bright pink by dinner. But the variant is present at substantial frequency in modern European populations. Around 60% carry at least one copy, according to some estimates, and it is a genuine legacy of interbreeding between our ancestors and Neanderthal populations somewhere around 50,000 years ago. The scientific interpretation is that when modern humans moved into higher latitude Europe, they encountered lower sunlight, colder climates, and different ultraviolet exposure than they had experienced in Africa. Rather than accumulating adaptive mutations from scratch, they picked up pigmentation variants from Neanderthals who had already been adapting to those conditions for hundreds of thousands of years. Neanderthals gave us a partial pigmentation package.
If you are a person with pale skin, freckles, prominent moles, and a tendency to burn, and if your ancestry traces to Europe, some of the reason is that your ancestors saved themselves an evolutionary shortcut by getting the local adaptation for free. Number 7. SCN9A, the Neanderthal genes that make some modern humans more sensitive to pain. Pain is one of the most fundamental sensations we experience. Sharp objects, hot surfaces, damaged tissue, all of them trigger signals through specialized nerve pathways that eventually register in the brain as pain. That signaling system depends on a small number of critical proteins, including one called sodium channel NAV1.7, which is encoded by a gene called SCN9A on chromosome 2. NAV1.7 is expressed at high levels in the sensory neurons that detect pain, and small mutations in the SCN9A gene can dramatically change how sensitive an individual is to painful stimuli. Some rare inherited mutations completely knock out NAV1.7 function, leading to a condition called congenital insensitivity to pain, where affected individuals cannot feel pain at all, with often devastating consequences, since without pain signaling, injuries go unnoticed and untreated. At the other end of the spectrum, some variants of SCN9A can make individuals significantly more sensitive to pain than average. In 2020, a team led by researchers including Hugo Zeeberg and Swante Pabo, the same partnership behind much of the Neanderthal DNA and COVID-19 work, identified 3 specific variants in SCN9A that had entered the modern human gene pool through interbreeding with Neanderthals. The variants are named after their positions in the protein sequence: M932L, V991L, and D1908G. A larger 2023 follow-up study, published in Communications Biology by a team led by Kaustubh Adhikari, investigated what those variants actually do in living people. The researchers recruited a large cohort of adults in Latin America, a population where Neanderthal inherited variants tend to be well preserved, and tested their pain thresholds using a controlled experimental protocol. Individuals who carried all 3 of the Neanderthal SCN9A variants showed measurably greater sensitivity to mechanical pain, being poked with a sharp object, particularly after prior exposure to a mild irritant like mustard oil. Their pain thresholds were lower. The signals from their sensory neurons were, in effect, tuned to fire more readily. Whether this heightened pain sensitivity was ever advantageous is an open question. It may have been an accident of introgression, a variant that came along when Neanderthals contributed other useful DNA to modern humans and simply hasn't been selected against strongly enough to disappear. Or it may have offered some subtle advantage in a prehistoric context, where a lower pain threshold could translate to faster protective reactions to injury before serious damage was done. Whatever the reason, some fraction of modern humans, particularly those with Latin American and Southern European ancestry, walk through the world experiencing pain a little differently from everyone else, because of a set of variants inherited at some point in the last 60,000 years from a Neanderthal. Number 6. TLR6, 1, 10. The immune genes that protect us from infection and probably also give some of us allergies. The innate immune system is the first line of defense the body has against invading pathogens. It doesn't rely on the specific antibodies produced by the adaptive immune system. Instead, it uses a small set of general-purpose sensors, receptors on the surface of immune cells, that recognize broad molecular patterns characteristic of viruses, bacteria, and fungi. A central family of these sensors is called the toll-like receptors, or TLRs. There are 10 different TLRs in humans, each specialized to detect different classes of pathogens. 3 of them, TLR6, TLR1 and TLR10, sit next to each other on chromosome 4 in a tightly linked cluster. And a substantial fraction of modern non-African humans carry a version of that cluster that came from Neanderthals or Denisovans. The discovery was made in 2016 by a team led by Janet Kelso at the Max Planck Institute for Evolutionary Anthropology, working alongside a parallel team led by Michael Dannenman. Both teams, publishing simultaneously in the American Journal of Human Genetics, identified 3 archaic variants of the TLR6, 1, 10 cluster that had entered modern humans through interbreeding with Neanderthals. 2 of the 3 variants most closely matched Neanderthal DNA. The third most closely matched Denisovan DNA.
Together, they were present at high frequency across modern non-African populations, around 50% of individuals in some European groups, higher in some Asian groups. Functional analysis showed that these archaic-derived variants were associated with increased activity of the toll-like receptor genes. That means immune cells carrying the archaic variants react more strongly to pathogens. In the context of a prehistoric world full of new microbial threats, many of which our African-derived ancestors had never previously encountered, that heightened reactivity may have been life-saving. It plausibly helped modern humans survive in new environments, from the Mediterranean to Northern Europe to East Asia. But heightened immune reactivity comes with a cost. Immune systems that fire more readily against genuine pathogens also fire more readily against harmless triggers: dust, pollen, cat dander, food proteins. The same variants that helped our ancestors fight off infections in the Levant 30,000 years ago appear to contribute today to elevated rates of allergies and asthma in populations that carry them. If you're allergic to something, and if your ancestry traces to anywhere outside of sub-Saharan Africa, there's a reasonably good chance that some of the reason is a Neanderthal, or a Denisovan, variant of an immune gene that decided that pollen counts as a hostile pathogen. The pattern shows up clearly in modern epidemiology. Rates of asthma, hay fever, and food allergies have risen dramatically in industrialized populations over the last century, a trend usually attributed to changes in modern hygiene, indoor environments, and microbial exposure during childhood. But the underlying genetic susceptibility to those conditions is not evenly distributed. It clusters in populations that carry the archaic TLR variants. Sub-Saharan African populations, which do not carry these variants, tend to show lower rates of allergic conditions even after controlling for lifestyle and environmental factors. It is not the only factor, but it is a factor.
They gave us disease resistance, and at the same time, they gave us hay fever. Number 5. The Neanderthal progesterone receptor, the gene that appears to protect some women from miscarriage. Progesterone is one of the central hormones of pregnancy. It is produced by the ovaries and the placenta, and it acts on cells throughout the body via a specialized receptor protein called the progesterone receptor, encoded by a gene on chromosome 11. Adequate progesterone signaling is essential for maintaining pregnancy. Insufficient signaling is associated with increased risk of miscarriage, particularly in early pregnancy, and can also contribute to abnormal bleeding. In 2020, Hugo Seeberg and Svante Pääbo of the Max Planck Institute for Evolutionary Anthropology, publishing in Molecular Biology and Evolution, identified a specific variant of the progesterone receptor gene that had been inherited by modern humans from Neanderthals. Then, in a much larger follow-up study, using data from the UK Biobank, which includes detailed medical and genetic records from around 244,000 British women, the same researchers investigated what carriers of that variant actually experienced in terms of reproductive outcomes. The results were striking. Women carrying the Neanderthal inherited version of the progesterone receptor were less likely to experience miscarriage. They were less likely to experience bleeding during pregnancy, and they tended, on average, to have larger families over their reproductive lifetimes. The size of the effect is not enormous. It's not the case that carriers were doubling their number of children compared to non-carriers, but it was measurable and consistent across a sample of more than 200,000 women. In the specific outcome of miscarriage risk, carriers of the Neanderthal variant showed a roughly 15% reduction compared to non-carriers after controlling for age and other confounding variables. The mechanism, as far as researchers can tell, involves the number of progesterone receptors produced by cells. Women carrying the Neanderthal variant produced more receptor protein per cell than women without it. That likely made their cells more sensitive to circulating progesterone, providing stronger reinforcement of the hormonal signals needed to maintain a healthy pregnancy. The variant is present at meaningful frequency across modern non-African populations. In some European groups, roughly 20% of women carry at least one copy. 20% is a substantial fraction of every population where the variant is found, which means that a real number of pregnancies successfully carried to term today and a real number of children who exist as adults trace some fraction of their existence to the fact that a Neanderthal ancestor happened to contribute a slightly better version of a fertility gene to the modern human gene pool 60,000 years ago. Neanderthals may have helped modern human women stay pregnant. That is one of the most quietly consequential inheritances any extinct species could possibly leave behind. Number 4. OAS1, the antiviral gene that Neanderthals contributed to modern humans and that appears to reduce the risk of severe COVID-19. If entry number 2 on this list is going to be about a Neanderthal gene variant that dramatically increases the risk of severe COVID-19, we should be equally clear that Neanderthals also contributed variants that appear to protect against it. The story of Neanderthal DNA and modern disease susceptibility is not one-directional. Neanderthal introgression pulled the genetic dice in multiple directions at once. In February 2021, Hugo Seeberg and Svante Pääbo, again, published a paper in the Proceedings of the National Academy of Sciences describing a Neanderthal inherited variant with a very different effect from the chromosome 3 variant they had previously identified. The variant is located on chromosome 12 in a gene called OAS1, 2'5' oligoadenylate synthetase 1. OAS1 is part of the antiviral machinery of the innate immune system. When cells detect double-stranded RNA, a hallmark of many viral infections, OAS1 activates an enzymatic pathway that helps destroy viral RNA and shut down the infection at the cellular level. The paper identified a specific Neanderthal-derived variant of OAS1 that was associated with a substantially reduced risk of severe COVID-19. Individuals carrying the variant were less likely to be hospitalized, less likely to require intensive care, and less likely to die from an infection with SARS-CoV-2. The size of the protective effect was estimated at roughly 22% reduction in the odds of severe disease per copy of the variant carried. The mechanism has to do with how the OAS1 protein detects viral RNA. The Neanderthal-derived form is longer than the standard modern human form, extending further into the cellular membrane, and it appears to be more efficient at detecting SARS-CoV-2 specifically. When the virus enters a cell and begins replicating, the Neanderthal version of OAS1 activates the antiviral pathway faster and more strongly. The infection is contained more quickly at a lower viral load before it can trigger the runaway immune reaction that causes the severe respiratory failure seen in the worst COVID-19 cases. The ancestry of the variant was confirmed by comparison with sequenced Neanderthal genomes from 3 different individuals, a 50,000-year-old Neanderthal from Croatia and 2 much older Neanderthals, approximately 70,000 and 120,000 years old, from southern Siberia. The match is as close to the exact as a long stretch of DNA can be by chance, or a shared ancestral trait. It came from interbreeding. The variant is present in meaningful frequency in some non-African populations, particularly in Europe, which means that when the COVID-19 pandemic swept through Europe in 2020, the outcomes of individual infections were shaped in part by which archaic variant a particular person happened to inherit. Some people got the Neanderthal variant that made things worse. Some people got the Neanderthal variant that made things better. Some people got both. The 2 variants sit on different chromosomes, so they are inherited independently, and their effects roughly cancel out in individuals who carry both. And the sobering fact is that all of this, the difference between whether a modern human patient in an intensive care unit lives or dies from a virus that emerged in 2019, was shaped in significant part by an encounter between 2 hominin species in the Middle East around 60,000 years ago. Number 3. EPAS1, the Denisovan gene that lets Tibetans live on the roof of the world.
The Tibetan Plateau sits at an average elevation of roughly 4,500 meters above sea level. Oxygen concentration at that altitude is around 60% of what it is at sea level. For lowlanders, spending extended periods at that elevation causes chronic altitude sickness, headaches, nausea, cognitive impairment, and in the long term, a condition called chronic mountain sickness, in which the body produces so much extra hemoglobin to compensate for low oxygen that the blood becomes dangerously viscous, leading to cardiovascular complications and pregnancy loss. Tibetans, however, thrive at those elevations. They do not develop the elevated hemoglobin response that lowlanders show. Their blood oxygen delivery works differently. They live, work, farm, and raise families at altitudes that make almost any other human population extremely uncomfortable within a few days of arrival. For decades, the mystery of how Tibetans do this was one of the most active questions in human high-altitude physiology. Then, in 2014, a team led by Rasmus Nielsen at the University of California, Berkeley, and Emilia Huerta Sanchez, publishing in Nature, provided the answer. The Tibetan advantage traces back to a specific variant in a gene called EPAS1, endothelial pass domain protein 1, on chromosome 2. EPAS1 is a transcription factor that regulates how the body responds to low oxygen conditions. In Tibetans, a specific haplotype of EPAS1 dampens the standard hemoglobin increase response to altitude, which prevents the dangerous blood viscosity complications that lowlanders develop. When Nielsen's team compared the Tibetan EPAS1 haplotype to genomes from archaic humans, they got a striking match. The Tibetan version of EPAS1 matched the sequenced Denisovan genome, the same species covered in previous episodes of this series, whose only physical remains are the small finger bone from Denisova cave and, more recently identified, a nearly complete Chinese skull. The Denisovans contributed the high-altitude version of EPAS1 to a population that would eventually settle the Tibetan Plateau sometime around 30,000 to 40,000 years ago. Some Han Chinese individuals, perhaps 1 or 2%, also carry the same Denisovan haplotype at low frequency because Tibetans and Han Chinese diverged from a shared ancestral population several thousand years ago. But it is in the Tibetans that natural selection has driven the Denisovan variant to near fixation, because on the roof of the world, having it means the difference between chronic illness and thriving. A 2021 follow-up study published in the Proceedings of the National Academy of Sciences worked out the timing more precisely. The Denisovan variant appears to have entered the Tibetan ancestral population early, but remained selectively neutral for a long time, until conditions changed and Tibetans permanently colonized the plateau after the last glacial maximum. Only then did natural selection begin driving the variant to high frequency. Additional studies in 2016 and 2024 extended the finding across the wider Himalayan region, Bhutan, Nepal, Sherpa populations, and showed that the Denisovan-derived EPAS1 haplotype is present at high frequency across a wide swath of high-altitude Central Asian populations. In one systematic analysis of nearly 1,500 individuals across Bhutan and Nepal residing at altitudes between 86 and 4,550 meters above sea level, 5 specific single nucleotide polymorphisms that characterize the Denisovan haplotype were present at high frequency, and their frequency correlated positively with the altitude at which the individuals lived. The higher the community lived, the more common the Denisovan-derived variant became. The physiological consequences are dramatic. Sherpa mountaineers famously carry Western climbers' equipment to the summit of Everest at altitudes where lowlanders develop pulmonary edema and cerebral edema. Tibetan farmers plow fields at 4,000 meters. Tibetan women bear healthy children at altitudes that would cause pregnancy complications in almost any other population on the planet. In every single one of those cases, part of the biological machinery making that possible was inherited from a species most of the world has heard of only in the last 15 years.
Every one of those individuals, millions of people alive today, is walking around with a fragment of DNA in their genome that came from a species most people have never heard of, whose only complete skull was just identified in a Chinese well in 2025. And that fragment of DNA is what lets them breathe at altitudes where the rest of us struggle. Number 2. LZTFL1. The Neanderthal gene cluster on chromosome 3 that has been called the single most important genetic risk factor for severe COVID-19. In September 2020, 6 months into the COVID-19 pandemic, a large international genetic association study identified a specific region on chromosome 3 that appeared to be strongly associated with the risk of respiratory failure in patients with SARS-CoV-2 infection. The region, called 3P21.31, contained a cluster of genes including LZTFL1, FYCO1, XCR1, and CCR3. Individuals carrying the risk variant of this region were approximately 3 times more likely to require mechanical ventilation and had significantly higher risk of dying than individuals who did not carry the variant. That in itself would have been a major finding. What Hugo Zeebarg and Swante Pabo showed in a paper published in Nature in September 2020 was that the risk variant had come from Neanderthals. approximately 50 kilobases of DNA at the 3p21.31 locus, including the risk-conferring variants, were essentially identical to the corresponding region in sequenced Neanderthal genomes. The match was too close over a stretch of DNA too long to have arisen from anything other than direct inheritance from Neanderthals through interbreeding approximately 60,000 years ago. The distribution of the variant across modern populations is striking. Around 16% of Europeans carry at least one copy. Around 50% of individuals in South Asia carry at least one copy. In Bangladesh, the frequency exceeds 60% in some sampled groups. In East Asia, the variant is almost entirely absent, a pattern that suggests strong selection against it at some point in East Asian population history, possibly during previous epidemics that we haven't yet identified. Something somewhere between 20,000 and 10,000 years ago appears to have killed off the East Asian carriers of this variant at a high enough rate that they largely disappeared from that gene pool. We don't know what that something was. The functional mechanism is still being worked out. The variant appears to affect the expression of one or more of the genes at the 3p21.31 locus, particularly LZTFL1, in a way that influences how lung cells respond to viral infection. It may also affect the CCR5 receptor, which HIV uses to enter cells, and interestingly, the same variant that increases COVID-19 severity appears to provide some protection against HIV infection. That may be part of why it persisted at high frequency in populations that carry it. It was selected against by one pandemic and selected for by another. When you look at the map of COVID-19 mortality during the 2020 pandemic, country by country, region by region, ethnic group by ethnic group, a substantial fraction of the variation in outcomes was driven by which populations had inherited this specific Neanderthal DNA segment at what frequency. There is no way to sugarcoat what that means. Some hundreds of thousands of people died from COVID-19, in significant part because their ancestors, 60,000 years ago, met a Neanderthal and had children. And the ancestors of the people who survived sometimes met the same Neanderthals and had different children. The pandemic that reshaped the modern world was not a fully modern event. Its outcomes were being shaped in part by an ancient meeting between two hominin species that both stopped existing tens of thousands of years ago. Number 1. The HLA immune system, the archaic gene complex that shapes how nearly every non-African human's body distinguishes self from non-self. The human leukocyte antigen, HLA, is one of the most important and complex gene regions in the entire human genome. It sits on chromosome 6 and encodes a family of proteins that sit on the surface of nearly every cell in the body. HLA proteins are essentially the way the immune system tells self from non-self. They present fragments of proteins from inside the cell to circulating immune cells, so those immune cells can check whether the fragments are from the normal proteins of your own body or from a virus, a bacterium, or a cancer cell that needs to be attacked. HLA is the reason organ transplants require immunosuppressive drugs. It's the reason blood type compatibility exists. It's the reason certain autoimmune diseases run in certain populations. It is arguably the single most important immune region in the human genome. And a substantial fraction of the HLA diversity present in modern non-African populations came from Neanderthals and Denisovans. The discovery, made across a series of studies beginning in the mid-2010s, is that specific HLA alleles, HLA-B*73 and several HLA-C variants in particular, that are absent from modern African populations but common in European, Asian, and Oceanic populations, match archaic DNA far more closely than they match the ancestral African variant. They came from interbreeding. In some populations, these archaic-derived HLA alleles are present at frequencies of over 50%. Papuan populations in particular carry high frequencies of Denisovan-derived HLA alleles, one of the strongest signals of Denisovan introgression anywhere in the genome. Why? The current best explanation is that when modern humans left Africa, they encountered pathogens they had never previously experienced. Local archaic populations, Neanderthals and Denisovans, had been living with those pathogens for hundreds of thousands of years and had, through the long, slow processes of natural selection, developed HLA alleles suited to fighting them. Interbreeding provided modern humans with a shortcut. Rather than waiting for our own immune systems to evolve suitable responses, we inherited pre-adapted HLA variants directly from local populations who had already done the work. The clinical consequences are still being mapped out today. HLA-B *73 in particular has been implicated in immune responses to a range of pathogens. Other archaic-derived HLA variants influence susceptibility to autoimmune conditions, where the immune system mistakenly attacks the body's own tissues. Conditions ranging from ankylosing spondylitis to celiac disease to certain forms of type 1 diabetes have HLA components that trace, in part, to archaic contributions. The immune system that keeps you alive when you get a cold is the same immune system that in some individuals attacks the joints or the thyroid or the gut lining. Both sides of that coin were partly written by Neanderthals and Denisovans. That inheritance persists. It shapes, today, which pathogens your immune system fights off successfully, which infections you're more or less susceptible to, which vaccines are more or less effective in you, and, when it comes time for it, how easily doctors can find a matched donor for an organ transplant. If you have European, Asian, or oceanic ancestry, some of the HLA proteins currently sitting on the surface of every cell in your body were designed, so to speak, by a Neanderthal or a Denisovan. And every time your immune system successfully fends off an infection, every day, silently, without you noticing, that inheritance is doing part of the work. Set every entry on this list side by side. Red hair on some heads, diabetes risk in some populations, a tanning response for people with European ancestry, sensitivity to pain in some individuals, immune reactivity that helps fight infection and causes allergies, a boost to fertility for some women, protection against COVID-19 for some individuals, and dramatically increased susceptibility for others. The ability of Tibetans to live at altitudes that would sicken almost anyone else, and the entire HLA system by which non-African immune systems distinguish threats from ordinary tissue. Every single one of these traits is influenced in some measurable way by DNA that came from an extinct species. The people who first proposed the possibility of interbreeding between modern humans and Neanderthals were for decades ridiculed. The conventional view was that the two species were too different, that any hybrid offspring would have been infertile, that our ancestors would not have been sexually attracted to Neanderthals, and that even if they had, no genetic trace would have survived to the present day. The conventional view was wrong on every single count.
Between 1 and 3% of the genome of every non-African human alive today is Neanderthal in origin. In some Oceanian populations, another 1 to 5% is Denisovan. These numbers vary by individual and by population. In some cases, they add up to more than 5% of your DNA. That is not trivial. That is a real portion of who you are biologically. You are not a pure member of any single hominin species. You are, everyone alive on this planet is, a mosaic. A record written in DNA of the specific encounters and specific interbreeding events that produced the specific combination of ancestors who eventually became your parents. Some of those encounters happened between Homo sapiens groups. Others happened, as this list has shown, between our ancestors and species that have not existed on this planet for at least 30,000 years.
The Neanderthals are extinct. The Denisovans are extinct. Whatever ghost populations produced the mystery DNA in West African genomes, those are extinct too. But they are not gone. They are, in the most literal biological sense, still here, living inside you, doing work in your body every day, shaping how you respond to sunlight, to pathogens, to altitude, to pregnancy, to pain. Some of it good for you, some of it not. Most of it, quietly, just running in the background of your life without you noticing. The extinct species on this list left no children of their own. Every direct Neanderthal, every direct Denisovan has been dead for tens of thousands of years. But they had children with our ancestors, and their descendants, their partial descendants, are still walking around, reading videos like this one, feeling pain a little differently, getting sick with slightly different odds, breathing in air at whatever altitude, with whatever immune response, with whatever pigmentation their inheritance happened to give them. They didn't disappear. They just spread out, diluted, absorbed into a larger population that carries them, unknowingly, everywhere it goes. Which means, in a way, that the extinction of the archaic humans was not really extinction at all. It was assimilation. They are inside you. They always have been. If you want to see more videos like this, subscribe, because on this subject, the science is still actively rewriting itself in real time, and we've barely started digging.